So thank you for joining us today. It's now my pleasure to introduce our speaker, Artur Stanchak, Field Application Specialist, Process Chromatography from Bio-Rad Laboratories.
Thank you very much, Alex, for that introduction, and thank you to the listeners for joining us today. Today we are going to be talking about strategies to address large molecule purification challenges. First, I will be discussing therapeutical entities considered as large molecules and associate with them challenges in the process chromatography. Next, I will be discussing how modern resins can address some of those challenges. I will give you an introduction to a modern strong anion exchanger, NuviaHPQ, followed by some application examples and a case study involving Immunoglobulin M. Over the last decade, large molecules gain a lot of interest as therapeutical products and open a completely new chapter in the process chromatography field. Some of the large therapeutical molecules also originate from plasma, and they are fractionated such as IgM and IgA. Other therapeutical entities include virus-like particles, viruses such as AAV, lentiviruses, and more. Plasmid DNA and other pegylated proteins can also exhibit some challenges in chromatography purification due to their size. Each molecule has their own characteristic, and depending on application it can vary greatly. Plasmids, for example, after fermentation vary in terms of yield and quality of supercoiled form. They required high salt concentration for elution. However, a solution for it is to use ceramic hydroxyapatite media with sodium phosphate as an eluent. Viruses can be sensitive to shear stress and have a large amount of closely related impurities. IgMs, on the other hand, are susceptible for aggregation and fragmentation in suboptimal conditions. All of them, however, suffer from slow diffusion and mass transfer kinetics, and as a result, a low binding capacity. In order to address the need of processing large molecules, a modern type of resin were developed. Some of resins' features are essential to achieve productivity goals. Here presented Nuvia resin family members such as Nuvia E'4A, a mixed-mode resins with hydrophobic and anion exchange modalities, and Nuvia HPQ strong anion exchanger are designed for handling large molecules. They are made of a very hydrophilic polymer, what reduces non-specific binding of impurities, and free up some binding sites to achieve high DBC. Pore size up to half micrometer, together with 50 micrometer bead size, narrow size distribution range are optimal for fast mass transfer kinetics. Even high flow rates won't impact the DBC dramatically because of the rigid polymer structure. This experiment was conducted to study the impact of residence time, hence flow rate, on DBC of thyroglobulin. Green bars represents 1.2 minute residence time and blue represents 2.4 minute residence time. Under tested conditions, NuviaHPQ, which is a strong anion exchanger, bounded up to 50 milligrams of thyroglobulin per milliliter of resin at 2.4 minute residence time. This outcompete especially agarose beads resins, which are softer and therefore more sensitive for high flow rates. Next example showing very decent dynamic binding capacity for fibrinogen achieved on a NuviaHPQ. As shown on previous slide, it was compared to agarose-based strong anion exchangers. You could notice that hydrodynamic radius is larger for fibrinogen than for thyroglobulin, even though the molecular weights are otherwise. In many cases like this one, not only molecular weight but also a shape of the molecule plays a very important role and should be carefully considered. In this study, 5 resins were tested for IgM binding. In this plasma fractionation project, the application was to bind IgM to deplete it from the feed stream, which also contains all other immunoglobulins. The goal was to increase productivity of the chromatography step. Nubia-HPQ showed much higher dynamic binding capacity comparing to other candidates at higher flow rate. Other resins candidate with dextran base matrix was able to achieve similar DBC, but only with 10-fold lower flow rate, 30 cm per hour. Implementing Nubia-HPQ reduced the operational cost due to higher productivity achieved by improved DBC and high flow rate. Now, please let me present you a case study that was done in our application lab located in Munich area. A tested material for this case study was the recombinant IgM molecule produced in cell culture. The aim for the study was to develop a non-affinity capture step. Quality attributes includes aggregation below 5% and purity over 90%. The purification must be cost-effective, therefore expectation for recovery was set to above 90%. Resin capacity target was set to 20 mg of IgM per milliliter of resin to reduce the cost of raw materials. The process also must be scalable and robust. As the first step, the DOE was conducted in order to learn about the design space. This is especially important if you are testing mixed-mode resins such as Nubia E'4A. Central composite design was selected and purification was done on commercially available 96 well plate, pre-filled with 20 microliter of resins per well. Two factors were tested, the pH and the conductivity. Different factor ranges were tested depending on the resin modalities. Nubia-HPQ is a strong anion exchanger, therefore typically, comparing to mixed-mode resin, it can operate in a narrower conditions range. From now on, all results shown are for Nubia-HPQ, even though other resins were also tested in this project. For this application, mixed-mode resins candidate required more development and fine-tuning work. Therefore, for simplicity and for robustness of the process, the Nubia-HPQ was selected. Here are counterplots generated based on DOE study. On the left-hand side, the monomer response showing that the pH is not a significant factor. Monomer content changes only with the change of the conductivity and remain very similar regardless of the pH change. For residual DNA content, banded response lines indicates there are some interactions between factors tested. In the next step, we switch from 96 well plate format to 1 ml pre-packed columns, which are also commercially available. As a first run, the middle points from the DOE study of the pH and the conductivity range were selected to learn how different formats affect results. Trace buffer system was used at pH 7.7 and conductivity was adjusted to 10 millisiemens per centimeter. On this slide, the same chromatography run is discussed as on the previous slide. Results for the size exclusion HPLC analysis showing that the pool 1, which was collected first, contains fragments. The second and the third pool contains majority of the target IgM, but the purity was not great at this stage. This observation supports hypothesis that smaller fragments have lower number of binding points comparing to the intact IgM and elute earlier in the salt gradient. Based on the DOE results shown before, the decision was made to increase the conductivity of the load material and the equilibration buffer. We learned that the higher the conductivity, the better the purity. By increasing the conductivity, the first peak, which appeared to be a shoulder of the main elution peak, is decreasing because higher conductivity does not allow to bind fragments to stationary phase. As a next step, the process was scaled up to 5 mm pre-packed column. The optimal conductivity from previous runs appeared to be around 19 millisiemens per centimeter. Only 20 mg IgM per milliliter of resin was loaded due to a shortage of this material. However, runs on 1 milliliter columns show no breakthrough with loading up to 30 mg IgM per milliliter of resin. Please note that factors such as binding conditions, flow rate, resin bed height might have an impact on the BC. Two pools were generated with and without a small shoulder on the standing part of the elution peak. The results for larger pool meet both quality and productivity requirements, being over 94% pure with over 91% recovery. Aggregates in this project were not an issue in a contrary to fragments, which tend to coalesce with the monomer IgM if suboptimal conditions are used. This slide shows analytical chromatograms from SEC analysis. Looking into a green rectangle, which represents the residence time for monomer IgM, there is a weak signal in the load sample and intense signal in the L8 pool sample. For the other two samples, flow-through and wash, the line is flat, indicating very low level of IgM in those samples. This nicely visualizes no breakthrough occurs and explain high recovery results over 91%. In summary, dealing with large molecules requires modern resins which are fit for purpose. From resins I briefly presented today, you can expect high dynamic binding capacity and productivity for large molecules. As mentioned before, this can be achieved because of the rigid hydrophilic base matrix, optimal bit size, and a narrow distribution range, open pore structure, surface extenders, and optimized ligand density. All of those Nubia resins features contribute to their high performance. With that, I would like to thank you for attending this webinar today. Please reach out to our team with any question you might have and visit our digital resource hub where you can request free resin samples, speak to our team, and more. I'm happy to take any questions.
All right, great. And as a reminder to our audience, you can go ahead and type in your questions for our tour now. So our first question is, what binding capacity levels can we expect for AAV applications?
Thank you for your question. However, before I answer it, it's important to acknowledge that there are many factors affecting dynamic binding capacity. This is true not only for AAV application, but also for others. In the recent AAV project, we observed a dynamic binding capacity in the range of 3 to the power of 13 particles per milliliter of resin. This was achieved for serotype 8. In this case, Nubia HPQ was used as a capture step, which is typically more demanding for DTC due to high level of impurities present in the tip stream. Binding conditions in this case were pH 8.8, conductivity was below 10 millisiemens. That was done in 1 minute residence time. In this case, impurities were removed significantly prior to the final polishing step.
All right, and our next question is, can you apply this purification workflow to GMP manufacturing?
Thank you for this valid question. Definitely. One of the most important features of UBHPQ resins, as well as other bioprocess resins, is the fact that they are easily scalable. From lab-scale screening columns to large manufacturing columns, you can expect comparable quality attribute results. Another very important aspect is the regulatory requirements for resins. Resins are classified as a critical raw material, and we manufacturing them in the controlled environment under a strict quality system. Each bioprocess resin is supplied with all required certificates, such as animal-free origin, DSA, TSA-free, as well as other comprehensive regulatory support file.
All right, well, it looks like we're running a bit short on time, so if we don't get to your questions, we'll be passing them along to Artur, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what if an ion exchange cannot be used in the purification process?
Thank you for this question. In some cases, strong anion exchangers might have limitations such as high conductivity of the load, for example. In such cases, I believe mixed mode resins would be a good alternative. I have in mind two mixed mode resins suitable for handling large drug entities. One of them is Nuvia A'4A, which is a mixed mode resin with hydrophobic and anion exchange modalities. This resin has also shown a high DDC level for serotype 8 of ABV, in the range of 10 to the power of 13 particles per milliliter of resin, thanks to its open pores. The second option is the ceramic hydroxyapatite type 2, which is widely used for VLPs, virus, and plasmid application. Its success is due to a unique mechanism of involving cation exchange and metal affinity modalities. If you are interested, please refer to our webpage where you can find more information regarding this, and you can request a sample to screen.
All right, well, thank you so much, Artur.
Thank you for joining, and please contact us with any questions.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.